Now we will study the Western blotting. Western blotting method is also known as sandwich method. Western blotting is also known as sandwich method or immunoblotting or protein blotting. It is used for the detection of protein. It also tells about the size and quantity of the protein, presence of proteins. It is also called as, same as ELISA, it is also called as qualitative or quantitative technique. This method is discovered by George Stark. This method is discovered by George Stark, but the name is given by W. Neil Burnett. The name is given by W. Neil Burnett, while the method is discovered by George Stark. The principle involved in Western immunoblotting is immunochromatography. What is immunochromatography? Is nothing but separation of sample, in this case protein, based on this molecular weight. Chromatography is separation of sample based on its molecular weight. So we use the principle involved is immunochromatography, and then the solid matrix is polyacrylamide gel. Sorry, it is not solid acrylamide gel. Polyacrylamide gel is used to separate high molecular weight and low molecular weight protein. Procedure. First sample is taken and it is placed in the gel. It is protein sample placed in the gel. The gel here we use is polyacrylamide gel. When we perform chromatography, based on its molecular weight, proteins get separated. And the separated proteins are transferred into a polymer sheet, and then it undergoes blotting, immunoblotting, and then they are detected. So the principle involves immunochromatography. So sample containing protein is placed on the polyacrylamide gel. Now separation is done based on its molecular weight. These separated proteins are transferred into a sheet, polymer sheet. This is a solid matrix and it is used for the blotting, that is immobilization of proteins. It is in Western blotting, immobilization of protein is done by sandwich method. After immobilization, they are detected by radiolabeled substances. Now procedure. Procedure involves tissue preparation, gel electrophoresis, transfer, immunoblotting and detection. First, we need to take a cell of tissue. A cell of tissue is taken. We add ice cold phosphate buffer saline and lysis buffer. We add both contain solutions, that is ice cold phosphate buffer solution, ice cold phosphate buffer saline, and then lysis buffer is added. Lysis buffer helps yield a maximum protein in the sample. This lysis buffer helps to yield maximum protein in the sample. And the next, after the tissue preparation, it is incubated in cold condition. Then some pressure is applied to the cell with a glass rod to make it rupture, and then gel electrophoresis is done. After transferring after placing the protein in the polyacrylamide gel electrophoresis is done. That is, electricity of 200 volts is applied for 45 to 60 minutes. Electricity of 200 volts is applied for 45 to 60 minutes. Then the protein molecules are separated based on its molecular weight from high molecular weight to low molecular weight. Transfer takes place. First, these separated molecules are transferred to polymer sheet. This acts as solid membrane. Then immunoblotting is done. It is done by following method. Firstly, to polymer sheet, blotting solution is added. Then it is incubated for 1 hour at room temperature. The excess blotting solution is drained and washed, and the polymer sheet is washed. Then again it is incubated for 4 degrees Celsius for overnight. Just before adding primary antigen antibodies, it is incubated at 4 degrees Celsius overnight. And then primary antibodies are added and washed with vigorous agitation. Then secondary antibodies are added, incubated for 1 hour at room temperature, and washed with vigorous agitation.